nice introduction. I hope the talk will be interesting and to the same level of the first two, of course. That were also very nice. So let's get started. So introduction was already there. I just want to give you a small introduction about our workplace, which might sound unusual maybe for most of the audience. We actually at Nazarbayev University, we're based in Astana, which is the capital of Kazakhstan. A quite new city, honestly, and a bit maybe tough to live in the winter because temperatures go down to -20, -30 degrees as common thing, but in the summer it's also quite hot. And our university, yes, microphone, okay, thank you. Yep. So our university is about 12 years old, so quite new one, and I've been working there for about 8 years. As the university is developing, of course, we now start to have more programs, as we started only from undergraduate programs. Now we start to have also masters and PhD across all the range of disciplines. And we also have research facilities that we built up in the last 6 years, and that are, of course, a little bit expanding and coming to the point of what the university should offer in terms of academic environment. These are some pictures of our group. Our group actually is focused on the development of optical fiber sensors and particularly looking at biomedical applications. So we are one of the first groups who's actually using technologies that are commonly used in distributed sensing, but into biomedical engineering. So we are looking at some of the biomedical applications and where such photonics technology can go to. These are some of our researchers. And these are some of our main activities. Our main focus right now is to work on cancer biomarker detection using optical fiber sensors, the detection of some viruses, for this also will be part of the talk of today, and to bring the detection in other liquids that are a little bit under-analyzed from the point of view of biosensors, but the interest is rapidly rising in terms of sweat, tears, and saliva. These are becoming some of the go-to analytes for current detection. And we also have the interest in some of the physical application and the combination of physical sensing and biological sensing, in particularly in distributed strain and shape and temperature sensing in very short medical devices. So we have the possibility of doing that. These are some of the many people that compose our group, but we also have many undergraduates and visiting staff as well. And these are some of our current projects that are funded. So in case, of course, some of you is interested to know more about the details of some of the specific projects, of course, feel free to stop me after the talk or in the next days or drop an email anytime. So let's begin. What I would like to cover in this talk is I will give an introduction about optical fiber biosensors, as this is a technology that is emerging, but probably has not reached the maturity level yet. And then what I would like to shift the attention onto is some of the minimalistic approaches that will make the fabrication of the devices much faster. So hopefully the fabrication will be faster and with simpler fibers, and that will bring to some low-cost type of devices that we can manufacture in scale, which is one of the challenges that still preventing us from reaching large market opportunities. So the idea is to employ this technology to detect viruses. So I would like to provide a brief overview about some of the current method detection for viruses and some of the things that the last pandemic, the COVID-19 pandemic, has taught us in terms of some of the detection opportunities. And then I would like to dedicate the last part of the talk to the case scenario that we have identified, which is the detection of pox viruses, as this is one of the case that has potential threats in terms of biosafety, and in terms of detection is harder than the conventional COVID-19 detection types in which the protein have different shapes. And then I will draw some consideration and conclusion. I would say more consideration and conclusion, as the topic, of course, is rapidly emerging, and there are not many conclusions yet, but rather there is a field that is developing into many branches throughout. So at first, I would like to start talking about optical fiber biosensors. And what I would like to start from, considering the technology in its nature, is a quote from Galileo Galilei, the famous Italian polymath and scientist, who used to say, measure what is measurable and make measurable what is not so. And this is a common point for people who are specialists of sensors and biosensors, of course. And we have known many technologies that measure what is measurable. So we have sensors that, of course, rely on technologies that are quite well established, piezoelectric, electrochemical, electrical, as well as some of the optical sensors, fluorescence, and many other methods that, of course, have reached a very large and popularity and also industrial maturity. And these technologies measure what is actually measurable. So, as a matter of fact, we have covered well this part. There is, however, another side of the problem that is what we try to cover. Of course, many people in this room are doing that as well, and it is the target of making measurable what is not so. And particularly, that comes to several of the environments that are not easily accessible. The human body is one of them, of course. Sensors that are able to measure in situ is a quite challenging task. Of course, sensors that are capable of measuring in some specific point with minimal invasiveness and many other characteristics such as biocompatibility, long-term measurements, continuous measurements, is a quite hard to achieve target in many applications. Optical fiber technology is a good technology to address this target because it has some of the advantages, opportunities that we can leverage on. So let's start from some of the definitions. In general, optical fiber sensors, which are often labeled as OFS or FOS, make use of an optical fiber or an optical fiber device as a sensing element. So the idea is the optical fiber is a waveguide for light. It basically traps light beneath the core of the fiber. And we're using that information or a device that basically is based on this information as a sensing element. The idea is that the fiber itself also is used to transport light back and forward. So in most of the cases, the sensor is simply a device within the fiber, or in some cases, we'll see it's not even a device. The whole fiber is the sensor itself. The optical fibers are, in general, small, lightweight, and minimally invasive. In general, the fibers that we use in our lab have a diameter of 250 micrometers, basically 3 times the size of a hair. They are biocompatible, and we have plenty of standards, particularly the ISO standard that allows us to say so with a certain degree of confidence. They are immune to electromagnetic interference. That means they can really operate everywhere. They allow remote sensing. The optical fiber is the waveguides that has the lowest loss among all microwave and photonic type of waveguides. And that means that with a loss of 0.2 dB per kilometer, we can virtually place our source of light and our sensor at kilometers of distance with nearly no effect on the propagation itself. And the other thing is that it has a small sensing region, which allows us to stack, for example, many sensors within the fiber itself. So generally, how we design optical fiber biosensors is, and sensors in general, is we try to have a sort of approach that integrates the application, the physical principle, but also the specification and the standards, particularly when it comes to biomedical devices. Some of these devices are designed to operate in situ, and therefore they have to have specific characteristics that makes them operative in this type of environment. It's also important to understand that some of the concepts, such as, for example, the interrogation of the device, that the hardware that we have to have in order to accomplish the task. In some cases, the hardware is simple, such as an LED light source and a simple photodetector, but in most of the cases, the hardware is a bit more complicated. And the hardware is only one part of the whole sensing system. The other one is the sensing unit, which for many applications, of course, has to be disposable. So we have to take into account also some of the cost, affordability, and operation characteristics that the sensors have to have. One of the key points of optical fiber sensor that is almost unmatched by other technologies is that we have the possibility of having sensors of different kind. In general, sensors as we know them, they operate detecting one device using one working principle. And that is done with an intrinsic device, so a sensor within the fiber, or a sensor that has external element outside of the fiber. And in this case, we label that as an extrinsic sensor. However, we have multiple approaches that enable to extend what we actually measure. For example, multiplexing is a common feature here. The fiber itself can host many sensors. And when I say many sensors, it means many sensors per centimeter. So we can have a really narrow distribution of sensors within the space. But also we have the access to distributed sensors. in which the whole fiber acts as a sensor. So each of these points of the fiber is a sensor in itself. And with the technology that we have, and I'm going to show today, the resolution can be as low as 10 microns. So it means that every 10 micron, we have a new sensing point. So that allows us for really a narrow distribution of detection, or even semi-distributed sensors, in which we have a distributed sensor, but located in a short length of the fiber. And in this case, even the spacing can be even below 10 microns. So we have technologies that allow even that. So that means that we can measure not just parameters, but even the distribution in space of such parameters. And the multiplexing is becoming quite common nowadays. And we'll see some of the protocols as well today that allow us to, for example, make a parallel detection of many sensors, but with a simultaneous scan. So where there is no multiple scan and there is only one source and one detector. So we're not duplicating basically the hardware. So in general, what I'm going to discuss is about some of the biosensors and some of the physical sensors, however, operating in the biomedical environment. In general, physical sensors measure physical parameters such as strain, pressure, and temperature, while biosensors based on optical fiber, they aim at measuring concentrations, usually of target molecules that we want to detect, which can be proteins, which can be viral particles, and in some cases, even cells. The measurement can be performed in three operative scenarios. One is in situ. So, for example, a fiber within the human body performing actually a measurement in blood. Or it can be done through a medical device or within a medical device, or it can be done, for example, around the body, as in the case, for example, of wearable sensors. And this applies, for example, to both types of technologies. So, for example, we have been working on some of the wearable platforms that detect in sweat, or they can detect, for example, in tears. And in this case, the sensor, at least the sensing device, can be worn. When it comes to the maturity of the sensing technology, these are some of the physical sensors that we have nowadays based on optical fibers. For example, the intra-Arctic balloon is one of the most established applications that has reached a commercial level a few years ago. And that's basically a balloon that is usually inserted within the aorta, in which there is a pressure sensor constantly working and detecting the pressure, because as soon as the blood pressure drops, the balloon is inflated, basically giving another pulse. In this case, it's used, for example, in patients that have a very low cardiac rhythm. For example, that's a very common detection of fractional flow reserve. That is a method used in the detection of stenosis, of blood, of obstruction in blood vessels, basically, in which a fiber optic cable measures the pressure differential prior and after and before the obstruction, and doing the ratio of the two pressure levels, it's possible to identify different type of stenosis and the direction in which the stenosis is applied. This technology, for example, are commercial, and there are companies that are already doing that. If I'm not mistaken, the fractional flow reserve has been, for example, acquired by some of the large medical players. If I'm not mistaken, it was Boston Scientific, but I might be wrong on this side. There are some of the other applications in which we actually are reaching a quite mature level. Some of them include, for example, the monitoring of physical parameters, such as temperature and temperature distributions during thermal ablation of cancer. Our group has done several works regarding this matter. They are used, for example, in some of the intracranial pressure detection. They are used in monitoring of epidural anesthesia. That is one of the applications that I think people will focus from now on, because it has been shown that consistently with this type of sensor, we can prevent some of the mistakes in epidural anesthesia. It has been used, for example, there has been some research in doing long catheters for colonoscopy and for gastroscopy, so to monitor the digestive apparatus using many of the optical fiber sensors, but within a single fiber. And these are some of the applications related to biosensors that we have actually established. And here again, the bar shows the technology readiness level, which in this case is not reaching more than 5, because the technology is a bit more emerging. It's not as mature as physical sensors. But as we can see, there are some of the applications that have been already developed and probably are going to reach the commercial stage soon. Some of them include, for example, diagnostic of lung cancer, percutaneous glucose sensing. This is going to be one important one, for example, for the detection of some of the diabetic diseases. The detection of circulating breast cancer cells has also been demonstrated. It has been also applied, for example, in prostate cancer biomarker, in the detection of C-reactive protein, in urology, and even in some bacteria type of detection. There has been a work, for example, that shows a certain degree of reliability in detection of E. coli bacteria using surface plasmon resonance sensor. So in general, optical fiber biosensors, they do detect either biomarkers, hormones, viruses, bacteria, or different type of analytes. The actual point of where actually detection becomes consistently label-free is the understanding of what is the size of the analyte with respect to the wavelength in which we are operating. Optically, in general, small analytes such as proteins, for example, they actually appear almost invisible to us, as in general they are very tiny. Instead, large analytes, for example, as like cells, particularly at infrared wavelengths, they are quite large, and therefore the optical propagation problem becomes much more complicated. So we have to understand how each of the analyte basically acts with respect to the actual light propagation. Considering that the optical fibers, particularly the ones that we're going to see in this work, are very small, and the light is confined within the core, which is also very small. So we have to take into account what actually happens around the core. So the label-free detection is the principle that has been mostly used, particularly when it comes to detection at infrared wavelength. In general, label-free detection is quite common for the detection of biomarker and biomolecules because it's simpler to develop, and we can split the development of the sensing device from the surface functionalization that we are going to use in order to make the device specific and sensitive to the specific molecule that we have into target. So we have several methods that allow a fabrication that is quite rapid and repeatable. And we have also biofunctionalization methods. Some of them are based through metallic thin films. Some of them are based through the siliconization of the fiber. We'll see them shortly. And some hybrid methods, which includes both parts. And the evaluation of performance is quite similar to some of the optical sensors, not optical fibers, such as surface plasmon resonance or SPR sensor, that most of the biologists have in their lab and play with as a daily routine. So as SPR works, some of the performances are expressed in similar terms as in his work. The other advantages of optical fibers is when it comes to specific type of detection is the ease and rapidity of fabrication, and we'll see some examples soon. They can operate in situ, they can also operate in vivo, and they are biocompatible. And hardware plays usually quite fast, so we have detection of a few seconds or even a few milliseconds, depending on the approach that we're using. So they are label-free. The method is encoded in the bioreceptor. We can split the problem of how we make the fiber sensitive to the surrounding environment, how we make the bioreceptors on the surface of the fiber, and how they actually play the detection roles and convert the signal into an equivalent detection of the fiber. And they have high precision. And when I have high precision, I mean they really have detection the limits that are very low. That is because, of course, the use of optical technologies, particularly the use of telecom hardware, comes with a precision that is unmatched by some of the spectrometers that we have at visible wavelengths. The label-free principle actually is what we are gonna study today and see, and basically is more or less listed here. In general, it starts from a refractive index sensor. So the idea is that incident light is sensitive to the refractive index. Now, this schematic here just show a simple light transmission. In this case, sensitivity is simply the reflection at the interface between two different media, which is one of the simplest approaches we can have. But there are much more complicated ones. When we actually functionalize the surface of the fiber, that becomes our first medium using bioreceptors. In this case, when they are actually performing a recognition of some of the elements around the fiber and immobilizing them in the proximity of the sensor, this signal works for us as a change in the refractive index. So that's why the two things are quite similar in terms of detection problem. And that's why most of our sensors are calibrated with different refractive index media, much like surface plasmon resonance sensors are doing. It is also quite important that what is the role of the medium? the underlying assumptions from a physical perspective of all the optical fiber biosensors is that the medium in which they operate is low loss, such as, for example, blood or urine or sweat, but not saliva, because saliva has particles that make the medium having a high scattering. And that's why the detection in saliva is much more complicated. And it's important that the skin depth of the medium, so how much the waves penetrate the medium, is actually quite large. So that means that we don't see the reflection coming from the immediate surrounding of the medium. That, for example, prevents us from detecting cells with this method. Because if a cell is immobilized in the surrounding, as the cell is quite large and has a much more irregular shape, the optical waves will rebound in the cell multiple times rather than just being reflected on the surface. The functionalization protocols that we have are basically similar to the surface plasmon resonance, but we have additional ones that wouldn't work in SPR that actually can work on some of the optical fiber biosensors. In general, surface plasmonic sensor require a metallic thin film, because the metallic thin film has the dual role of vanishing the surface plasmon wave into a current that usually is transported and is vanishing from the sensor. And that's the order of reflection that usually is missing and that we are detecting in the sensor. And they host the bioreceptors. So a metallic thin film is good enough to host most of the bioreceptors. There has been sensors that have been reported using the antibody-antigen principle. That is the most important one, as well as aptamers for the detection of aptasensors. And in this case, the bioreceptor is an aptamer that usually either the antibody or the aptamers are studied separately. So we use other immunoassays. For antibodies, for example, it's quite common to use ELISA or Western blotting, while, for example, for aptasensing, it's much more common to use SELEX, for example, for the selection of aptamers, or methods that actually are quite more common within immunobiology for the selection of the appropriate bioreceptor. The other way that we can use is to use acetylization. So to silanize the fiber, basically using silane groups. That is quite typical functionalization, for example, for the use in some of the medical devices based on stainless steel that actually are not meant for detection, but rather, for example, are meant, for example, to extract some of the specific cell samples, particularly in the recirculating cells problems. In this case, APTES is based on usually cleaning the surface of the biosensor, performing a treatment with this sort of biological glue that allows the bioreceptor to stick very well. In general, there is an incubation in glutaraldehyde, in GA, and then the immobilization of the antibodies, and the surface treated with the APDMS or APDS with the excessive silane group is actually a good method for the detection of, for the immobilization of bioreceptors and then further the detection. So let's discuss some of the biosensors instead that we can find. This is an important point for what comes next. In general, when it comes to the optical fiber biosensors, we can divide them into two separate classes, each one of them with the pros and their own problems. The first type of class that I think is the most common one nowadays is those based on multimode fibers. So usually a fiber with a large core that makes it very easy to develop a biosensor around it because the fiber has a very large core and therefore it easily couples waves in and out of the sensor. Of course, plasmonic sensors are the most common one. As a matter of fact, a plasmonic fiber works by itself. If you take a large mode fiber and you do the position of a thin film using sputtering with appropriate thickness of the film, the sensor already has usually a plasmonic mode at visible wavelengths. And then you can detect that and tune it in order to match the wavelength that you want in your spectrometer, and that works. The problem of this method, however, is that they usually work not in reflection, but they work in transmission. So you need to place light source, sensor, detector. And that's not so practical. You would like the sensor to work in reflection because at the end of the day, you want to build a probe. So something that, for example, you can introduce in situ rather than having to fold the fiber to run forth and back. You might place a mirror after the sensor, but first of all, that's an additional element that you're using. And depending on the distance between the mirror and the sensor, some of these techniques don't work. So that's a quite tight optimization game. A similar principle is used by U-bend fiber. So that means like you need to bend the fiber very tightly in and out. And the fiber again has to be multi-mode. So these principles in general are quite simple. The sensors are effective. They have a usually high sensitivity. But what's the problem of multi-mode fiber? That is that in general, the sensor is not stable over time. That is because when you couple hundreds of thousands of modes into a fiber, the modal distribution changes over time depending on temperature, strain, vibrations, and all the other effects that you have. Even when you actually consider a propagation only a few centimeters or even few meters, it doesn't really maintain over time. Some of these modes are highly evanescent, and some of these modes are well coupled. So as a matter of fact, these sensors have a high sensitivity, but when you actually make a biological detection, the performances are sometimes suboptimal. That's why SPR, for example, is used as an alternative method for benchtop analyzing, but it's really not yet used in situ all the time. So the alternative is to use single mode fibers. In this case, there is no problem because there is only one mode. So at the end of the day, all the power is propagated by one mode. And the other advantages of single mode fiber is that there is a whole set of devices that you can use. For example, gratings. The previous speaker, for example, was introducing a Bragg reflector as a reflector that basically is created by multiple layers, have different refractive index and at a specific distance from each other. Such device would work only with single mode fiber. It wouldn't work in a multi-mode fiber because each mode will have a different effective refractive index, and therefore each mode will see a different distance between the layers, and therefore each mode will reflect a different wavelength. And since you don't control the modes, you will have a sort of mess in terms of detection. Also, interferometers have the same principle. You can make sensors combining one or multiple wave paths that run a different phase, and they need to be always having the same refractive index. An interferometer in a fiber with two modes will basically behave as a two-wave interferometer. But usually when you have hundreds of thousands of modes, the interferometers don't work. So this is one of the advantages, and single mode fibers, of course, have a very high precision. Additional advantages are the fact that single mode fibers usually are used in telecommunication. So they are exactly the fiber that has been used, for example, in long-haul transmission systems, such as the one that you have, between multiple cities, the backbones, urban backbones, urban and long-haul type of transmission system, as well as the ultra-long-haul ones, such as the one running under the seas and under the oceans. They all share the same fiber that we know as SMF28, that is being standardized with a certain core and cladding size. And in general, all the industry already gives us this standard to work with. This fiber has an exceptionally low loss, so that makes it extremely usable even for long distances, which comes handy when you actually plan to detect viruses in urban or indoor environments. And this fiber also, of course, has only one mode. So it kind of comes quite cool. The problem of these sensors, however, is that the fabrication is quite hard. For a grating itself is not a biosensor because it's still not depending from the refractive index. So you got to change the grating design in order to make the sensor suitable for biosensing. Some of the schematic that you see here, like long period gratings or tilted Bragg gratings, they do that, but they have another disadvantage. They need to use modes that are in the fiber cladding. So the fiber cladding is multi-mode, and again, you have the same propagation of multiple modes within the fiber, and that makes the sensor a little weaker in terms of performance. Interferometers have an exceptionally high performance when it comes to sensitivity. So they are really high level technology, but they are very hard to make. Making, for example, a microfiber interferometer is quite hard. So that means that it's easy, for example, to get us one sample and discuss it, but it's hard to make a repeatable fabrication of these interferometers over many samples when it comes to refractive index sensing and biosensing. So what we did in our lab was to try to develop more minimalistic type of fabrication. So an approach that simplifies to the extreme the fabrication of the sensor, where basically any researcher with a few hours of training can come and fabricate as many sensors as they want. But of course, it has a drawback of having maybe some harder type of detection, and still based on a single mode fiber. We've done a little bit of radar plot here that shows some of the characteristics of the sensor that I've described before. So in terms of ease of fabrication, suitability for automation of the fabrication process, that is the most important one when it actually comes to scaling fabrication processes, sensitivity, limit of detection in at least one biological detection application, and readiness in terms of commercial outcomes. And predictably, these are some of the minimalistic approaches that we have seen. Actually, of course, they are quite good in terms of fabrication and automation. There is one here missing, but I will present soon a paper in a couple of days that will show how you can even have a zero fabrication process here. So there is really no other application than the only necessary one that you would have. And yet you have as a working sensor, and actually a pretty good one. But we still don't have, of course, yet a high level of readiness, and that's where is our next goal. Some of the gratings type of sensor actually have demonstrated quite good readiness. Plasmonic gratings, for example, have been applied, and there is even a commercial company that is industrializing them right now. But yet their fabrication is not so scalable at the moment, because you have to make a grating, then you have to make a plasmonic layer. The plasmonic layer has to have exactly the right thickness, otherwise, etc., etc., etc. There is plenty of steps that has to be done. Interferometers also depend on the point of sensitivity. Usually the structures are extremely high sensitive. And that's of course positive, up to the point that in some cases they're even too sensitive. But yet the problem is the fabrication. That is extremely hard. So you don't really make thousands of them. You only make few samples. So let's go and get some more look into some of the minimalistic approaches for optical fiber biosensors. So, which is, of course, the first part of the previous chart. What we have identified, and I will discuss, is three methods. One, that is a device that we have called ball resonator, that is a reflector fabricated on the fiber surface that has a very tiny reflection, but somehow it works and has basically a tiny reflectivity, but detectable, and coming directly on the fiber tip. Another one are some of the reflectorless sensors, so sensors that implement the same detection method using gratings, but without a grating. And then we have developed just in the last few weeks a new approach that is called semi-distributed biosensor, but I will not discuss that into this presentation. We have a presentation on Tuesday, so if you are interested, of course, I would like you to attend it as well, and you can see the structure more in details. So the ball resonator sensor that we have used, okay, has an extremely easy fabrication. It basically consists of making a sphere, such as this one, on the surface of a single mode fiber. This fabrication, if you have the right splicer with the rotation mechanism, is extremely simple because it's been already done in diffractive lenses. That is exactly the same recipe that you use in diffractive lenses. The main difference is that while diffractive lenses are based on multi-mode fibers, this one is based on single mode fiber. From an optical perspective, there is, of course, a huge difference because now you have to analyze the reflections back into the fiber. But in terms of fabrication, the job is already done. So it takes about 30 seconds to make one. Just when you load the fiber into the machine, most of the job is taken care of. The main difference that we have and what makes it attractive for biosensing is that while generally you need another device to excite the resonator, and in this case it works as a whispering gallery mode or WGM, such as, for example, exciting that with a taper, or as in this case, having a light, a detector, and exciting some of the modes that resonate around the sphere. That is quite hard. And of course, this is in free space. This is in free space with two devices. This one is directly into the fiber. So we just launch the fiber into the ball and see the light that comes back. But the fabrication is extremely simple. here are the steps, but basically what you do is simply to load 2 fibers into a splicer, and when you have the right recipe, the job is done. In our lab, we already have, of course, this setup, so for us, it's actually quite easy to align. And these are some of the samples that we have fabricated. I just rendered a mesh that shows basically the device in 3D. This is actually how we interrogate the device. So we take an analyzer, this one, that is used already in distributed sensing, and we monitor the spectrum at the tip of the fiber. So we set the distance at the tip of the fiber, and we monitor the spectrum right there. And these are some of the spectra and the way they change with different refractive index values. The first thing that comes to mind is that although these are 4 very similar devices, the spectrum is totally different from device to device. And that's one of the common points of working in these approaches. The spectrum is almost random. So you gotta have to deal with a random spectrum. But the sensitivity is quite large, and it goes in the sense of usually up to several tens or up to a few hundredths of dB per refractive index unit when actually you track how the intensity changes in each point. We're now having a student who is taking care of the software, so that what we are going to do next is, based on the many devices that we have fabricated, we'll just have a method that, regardless of how the spectrum looks like, it will take care of that. But it's not ready yet, so I cannot really show that to you right now. So here is how a Ball resonator works in some of the biosensing applications that we have developed. The first one is the detection of a cardiac marker, thrombin, to be more specific. That's a common case for detection because it's a protein for which we have plenty of knowledge. And we have biofunctionalized Ball resonator with a thrombin binding aptamer, so that's an apta sensor. And as the thrombin is actually bound on the surface of the fiber, that changes the optical spectrum. So what we have here is, in this case, we monitor a wavelength shift. Most of the time, we monitor an intensity change, and that actually progressively changes. So we could test basically the concentration dependence and the specificity, much like it's done on other biosensors. We have particularly used this Ball resonator for the detection of CD44, which is a cancer development factor that we are investigating. We have currently a project that is about the importance of CD44. And particularly, this is overexpressed in a subset of cancer stem cells. So it's quite important because it, for example, can enable us to detect and approach some of the breast cancer stem cells that we would like to, of course, study. So what we did basically is that we did a similar system, the Ball resonator, using a functionalization that we have done, and that's a quite standard one. And we actually place a sensor right into a tube, so that actually is mimicking actually an analysis. And here is how the detection performs. So we have done here multiple sensors. We have 3, but in reality we have much more than 3 to show that we have a dependence from the concentration of the analyte that is quite log linear. So for an exponential change of the concentration, we have an incremental change of the output, which here in this case is normalized by the sensitivity of the sensor. So the sensor is extremely precise because, as you can see, even concentration of few attomolars already give a response. The negative part is that, of course, it's operating on a wide range. And that's quite common for optical fiber biosensors. So it's not the only one, but actually almost all the works, except plasmonic ones that you see, have this type of log linear pattern. It's common in some of the electronic sensors as well, but not many. So in general, not on such a wide range of detection. And for example, this is how the sensor works over time. So it's binding quite rapidly. So it takes really a few minutes to have a detection, and the sensor actually detects. So the concentration, each step of increasing concentration is detected, and the sensor actually has a quite low detection limit. Again, we have tested the specificity using methods that are quite standard. And we even tested the response in different media, such as in serum or in PBS, to evaluate basically the differences. And in general, the sensor tends to have a log linear pattern in serum, but quite saturated pattern in PBS. Again, that's quite common in all optical fiber biosensors. So it's not a perk of this one, but it's quite common even on the methods that are not so minimalistic in terms of fabrication. What is quite interesting also is that we try to play on the functionalization to see if we can make the detection better. And while these two curves right here are the previous curves that I've shown before, these two additional curves show a device made in this case with metallic thin films rather than the APTES functionalization. And in this case, using again APT-MES, but the addition of gold nanoparticles after the functionalization. So the increase of reflectivity is quite remarkable. It's seen in the spectrum, which is not displayed here, but of course that's quite predictable. What we have also here is that the change of intensity is also quite remarkable. So the sensor is more sensitive. The problem is that this is a quite small data set. And while for these sensors we have many, many replicas, for this one we really don't have that many. The introduction of nanoparticles makes the sensor somehow less repeatable. So we have sensors that in general are very sensitive, but the sensitivity might be very different from sensor to sensor. And I don't know how this is actually beneficial when it comes to some of the applications. Now, a very important point is multiplexing. So I say that multiplexing is a key, but how can we do multiplexing in this kind? We have one source and one detector. So one possibility is to split up the detector, so to have one source and multiple detectors. That's, for example, what SPR scientists are doing. They make a parallel detection where they have one source, usually a chip that actually splitter and splits into multiple channels, and at the end of each channel there is a detector. Okay. The problem is that we operate with random spectra with a low sensitivity. Our detector is expensive. We do not really want to duplicate that. So we would like just to have multiple sensors, but one hardware. So what's the way to do that? Well, we can benefit from distributed sensing. As I said, distributed sensing analyzes the spectrum in every part of the fiber. So why not spacing each sensor? Here we have done with 4 sensors, but in our lab we have done with 16 sensors, and it kind of works in the same way. But each one of them appears in a different part of the fiber. So we have here a network of splitter that actually you can simply close into a box that has a different length for each of the fibers, and such that each of the sensors appear in different parts. So basically introducing a delay line in each of the elements, and it works. So now we don't have any more one sensor, but we have 4 sensors operating simultaneously. As I said, we have done in our lab up to 16. So, but virtually, considering that our analyzer can go up to 32 meters, and the sensors need to be spaced by 20, 30 centimeters at max, and or more, well, you can make calculation that hundreds of sensors virtually is possible. And as you can see, each sensor detects separately with a minimal crosstalk. So it works. And as a last element of how Ball resonators can work in practical application, this is a project that we have done where we have incorporated basically a Ball resonator into a wearable device shaped of like goggles that actually have a small channel and in which it can basically collect and flow tears. So the question is, can we make a detection in tears, considering that tears is actually liquid in which the volume is very small? The answer is actually yes, we can, particularly considering that tears are small, but if you compare that with the size of the fiber sensor, they're not that small. So at the end of the day, we still have the condition for detection. We have done this project with the idea of detecting some of the lipocalin 1, which is a biomarker for diabetic retinopathy. And we actually have a sensor that kind of responds quite well. So for each type of, notice that on each chart, the spectra are different. That's what I'm saying. This as a random spectrum. On each type of concentration, we have actually quite changing type of response. And again, with the same log linear pattern, that's because tears in general are not that dissimilar in refractive index from PBS. And the sensor can operate even dynamically. If you notice, we have a quite good staircase type of response, where each concentration step is almost instantaneously assimilated by the sensor. So when we go from PBS tears and then rising with the marker concentration, the detection is quite accurate. The other technology that I think could be very useful to discuss is what we have labeled as reflectorless biosensors. Now, reflectorless means we don't have a reflector, but yet we're still analyzing the reflective waves that come to the sensor. So where the waves come from, what actually we are detecting is the Rayleigh scattering. So we are detecting the Rayleigh scattering in every section of the fiber. There are methods quite common to make the fiber sensitive to the outer environment. One of them is etching the cladding away. Another one is tapering the fiber. These are methods that are quite established. But why they have not been used? The main problem is that when you start to have a fiber that is very sensitive and therefore very responsive to the surrounding environment, you also have high losses. So you need to tolerate high losses. The problem is that single mode fibers are engineered to have extremely low losses. So they scatter very little. So when you actually take a very small amount of scattering and you drop that down by 10 or 20 dB, it takes about 20 dB to make the sensor quite sensitive. You really fall below the noise of the instrument. So you can't really detect. The margin is about 6 dB, if I'm not mistaken. So how do you do that? What we actually used here is a fiber that has the core doped with nanoparticles. So it seems that the fabrication is quite complicated, but in reality, that is entirely done in the preform of the fiber. When the fiber is thrown, it forms several defects in the core of the fiber at the drawing point. But after the fiber is drawn, it actually looks like a standard fiber. So you can splice it like a standard fiber. You can use it like a standard fiber. You can fabricate hundreds of meters, and that works. So now we have a high scattering content, and we can tolerate these losses. So if you just take one of these fibers and simply etch it or taper it, actually you can make a biosensor. These sensors already exist. They are based on gratings, but here there is no grating. So the spectrum is the Rayleigh scattering. And as the Rayleigh scattering is a totally random signal that is different from point to point, you can really assume it as a additive white Gaussian noise. So if you take a white Gaussian noise generator, you will get spectra that are very similar to the Rayleigh scattering. So, and that's how it works, but it actually performs a detection. Our first take actually was this work where we actually performed a fiber, etched it. So did a metallic thin film deposition, exactly the same functionalization that has been developed for the Tosi biosensor on the same detection principle. And as you can see here, the nanoparticles that we have developed are magnesium oxides. That's why the fiber has an excess of magnesium on the surface when you do the surface element analysis. And the way it works is that by making the fiber thinner and thinner and thinner, you get a better sensitivity. So I'm not going to discuss too much this work into details, but actually it's a known method. The problem of this method is that it is quite demanding in terms of etching. So you have to etch the whole cladding away. Is there a better way to do that? One way is to use a taper. A taper takes about 25 seconds to be made on the right splicer. And it's the same one to make the ball resonator, by the way. So you can really catch two birds with one bean. That means that here, as you can see again, that device is quite sensitive. It has about 20 dB of losses. Notice that this is the launch fiber. This is the noise flow of the instruments, that's 6 dB away. So I'm not lying when I say that if you don't have a fiber with a high scattering, in this case, notice that the scattering is about 43 dB away, but some of the fibers that we have has about 50 dB of scattering more. So that a fiber engineered to scatter 10,000 times more than standard fiber. So obviously, that's quite an engineering process there. And here we made a taper. This taper has a diameter of 30 microns. So this is not a fiber with 1 micron diameter, as you find in some works that really try to get the diameter down, but however don't have a very high reliability. These tapers, they survive when actually you make a biological measurement. And this is how you detect it using distributed sensing. Notice this is distributed. So we get a wavelength shift in every single point of the fiber. This chart has 10 micron resolution. So when you approach the center of the taper, the waist of the taper, you get more sensitivity. And when you mark that point, you can see how the wavelength shifts in this point, you can get basically a detection that is quite accurate. This method is still developing, but I think it's a good one because it's also quite fast in making. So that's actually about optical fiber biosensors, minimalistic approaches. But what can we say about viruses? Why can these fibers be used for detection viruses? And what is the current method that we have? So I've made a small digression, and I would like to be apologizing in advance in case some of the data, I'm just an engineer. Of course, detection methods for viruses are quite complicated and established. I tried to gather, however, the most recent one, particularly as well, you all know, there has been a quite particular interest for detecting viruses in the last 3 years. So one of the important factors that it's been under consideration is where to detect the actual viruses. So this group actually was one of the first ones that tried to plan for a virus detection in wastewater. The key idea is that wastewater tend to collect a lot of things in their inside, which comes, for example, from households and so on. There is a parallel line of research that takes into account the detection of gray waters, such as, for example, in indoor environments, such as in a plane, the water that comes from the laboratory. So the general idea is that in case there are specific viral particles, that's a place where you can find them. We'll see there is some significant research addressing this thing. So the method of detecting viruses that is quite common nowadays include the PCR. The PCR actually is a chain reaction that takes a lot of time to develop in comparison to real-time detection techniques that, of course, have a different approach, but it's quite reliable. So the idea is that PCR tests that you have right here actually do perform a series of operations that is in general parallelized, but to give not real-time type of answers. The analysis of viruses in wastewater, on the other side, has been quite deep. This work, for example, has performed a detection in wastewater in several samples. Particularly, they detected some of the noroviruses, some of the adenoviruses, and some of the astroviruses that we have right here. All of them in different concentration ranges. One of the problems is that when you actually need to explore for a detection environment, to have such a narrow type of detection, such a narrow one order of magnitude or even below then, is usually not that viable. Many of the sensors saturate in general right here. Now you start to see one of the advantages of the optical fibers, that it actually detects across a wide range of concentration. So it's a drawback for some things, but for some other things, such as exploring in this environment, it actually can come as a quite handy tool. The conclusion of this study was that in general, there is a lack of methods that resolve multiple viruses within a single sample, that PCR standards were, back to 2018, when the first study came out, becoming the golden standard, but yet not instantaneous. And that when it comes to wastewater, you need some process control to be able to assess the virus recovery during the detection. Several works have been released a bit later for actually what could be the possible way to detect viruses into wastewater. I marked down this one because I think was quite a good one. It made a nice review about what could be the actual sample processing, so the virus concentration methods, how to collect the virus in order to basically get the concentration right, and how to perform the virus detection in sludge. So the idea is how to prepare each sample of virus in order basically to have the right preparation for each sample. So what the study did was try to spot up also in which sample of wastewater you could make the detection. Influence, so getting into the wastewater, effluence, so out of the wastewater, or basically during the primary, secondary, and tertiary treatment of the wastewater. So really we have now a sort of roadmap that in a urban type of environment can allow us to mark down where to make a detection. So if we were to have a sensor that could make a detection, I think nowadays we probably have the know-how to know where to place it. Here is some of the outcomes of the study, which basically collected several samples into wastewater and analyzed. There are several types of viruses. There is rotaviruses. There is, of course, SARS-CoV-2, which has been one of the ongoing viruses at the time. This study was published at the end of 2020, so when the pandemic was still ongoing. And here we have, for example, some of the adenoviruses. that's this one here. The adenovirus particularly have a quite large range of detection. But notice that the best performances in general, when it comes to detection limits and width of detection, are in the influent. So the idea is that the sensors need to be placed at the entry point for wastewater. And that, of course, is a disadvantage, because in terms of variety of viruses, you would like to detect towards the effluent side. The question of the study is knowing that when it comes to the green part of the chart, and therefore we are moving in general to the right part of this detection chart, if we had the right sensor, would it be also detectable enough? Okay, that's a question that is in the air because we still don't have data to back up this question. And I want to mention also this work that was reported in 2020. That, however, I think is a kind of a pivotal work to understand how to deal with that type of detection for the future. This is a chart that reports basically the number of titers that were found in wastewater in 4 different counties of Massachusetts, per each day. So they got samples of the wastewater. Actually, that was done before the pandemic started, but their study started differently. And you notice that after the pandemic started, the number of samples that were obtained was quite high. So it means that the wastewater samples do actually contain viral particles. So the feasibility is feasible. We need now to get the technology on how to do that. I just marked down another study, will not discuss all in details, which, however, is quite interesting because it points out some of the analytical tools that we have divided in systems that are based in tubes, so lab on a tube, lab on a chip systems that, however, require the virus to be collected and processed, lab in a box system, and even, I guess, lab on a drone system or lab in a cartridge system will be left for later. Particularly lab on a drone is quite hard because detecting viruses in the air is usually quite hard. So what can we learn from that, and how can we use, for example, these for some of the, for example, COVID-19, we have some data because now the pandemic has luckily finished. So we do have now some technological data. So apart from a few works that I've chosen one right here that were able to detect the virus in the air with the breathing system, notice, however, that this means an analysis of the breathing content to be done in, imagining that in real time. That's a quite hard device to be worked because it's not like a wastewater where basically you keep leaving and you just collect samples, but it means that you have to have an analytical tool that is placed and you have to breathe inside of it in order to make the analysis. These are some of the viruses that were detected in the air. There is not really yet a lot of methods that perform this detection in a faster way. There's been some works, however, that perform this detection using Apta sensors, either on chip, such as this work that was presented in 2021, and is based on surface plasmon resonance. And here is some of the results, basically, where there was a general concentration and a wave and shift observed. Although this work is quite preliminary, it was probably the first one to be done in 2021. There is some works that use plasmonic fibers, particularly fibers that have been etched on one side to increase sensitivity. And this work actually is probably the most analytical one when it comes to actually performances nowadays. So this was presented in 2021 and showed how a D-shaped fiber actually has spectra that really can change when multiple viral particles are detected. The authors did the study on SARS-CoV-2, and they compare with MERS virus, okay, which is another virus affecting respiratory apparatus, much as coronavirus. What is the actual problem and features of coronavirus and why it does not really apply to other cases? The main problem is that the coronavirus, as you can see right here, is directly in the structure. It has a spike protein that is all around the surface. So for detection with an optical method, it's quite simple because first of all, it's a spike protein which binds quite well, and antibodies have been already identified for this type of sensor. So the possibility of making the antibody antigen binding works quite well. The problem is that coronaviruses in general, not only this one, but in general, all coronaviruses have a crown. This corona actually is a crown that actually goes all around that has a lot of protein available for binding. We have seen from the previous slides that optical fiber biosensors work extremely well to bind the proteins. And it's probably the best thing that they do. So the question is, can an optical fiber detect the virus by detecting the protein that is available on the surface of the virus? That's the question. Let's see if we have an answer for that so far. So one of the case scenarios that we identified is pox viruses. The idea of detecting pox viruses rather than, for example, coronaviruses is that in general, pox viruses, such as variola virus, it's the causative agent of smallpox, is one of the deadliest ones that we have nowadays. We have a vaccination campaign, of course, that eradicated this type of virus, but it still exists. If I'm not mistaken, it exists in the USA and Russia in 2 separate laboratories. And there's been a work back to 2018 that was, I think, a bit controversial, that reported the reconstruction of basically horsepox virus from an extinct virus using only artificial intelligence. So that work a little bit caught the eye because one asked, okay, if that was done to horse virus, it was an extinct virus, could it be done on the also on smallpox, that of course is a lot deadlier than horse virus. And of course, has the virus such a deadly potential, the detection that with instantaneous methods takes a lot of interest. So what we actually tried to do is we developed a small case that we could handle in our lab using vaccinia. Vaccinia is one of the virus of the pox virus family. It can be found and detected either in body fluids, such as respiratory viruses, but also as wastewater in the environment. The idea is, can we detect the virus by detecting the proteins that constitute the active domains of the virus? And we have basically for this virus 3 proteins that we have identified: L1, A27, A33. There is a 4th one, B5R, but unfortunately we don't have the data ready yet to be shown at the time of this talk, but hope we have them soon. So for these proteins, actually the sushi domain is available and they are quite well known. So that could be the first step to detection of pox viruses. So the idea is we can try to do that. The results could be quite well portable since the active domain of the smallpox and vaccinia are quite similar. So these are some of the current methods, but a little bit faster. I just wanted to point out what is the structure of the particle. Notice that this is not like a coronavirus. That's a drawing that we made. The protein that we have available for binding on the surface of the viral particle is much smaller. The particle itself is larger, still smaller than the wavelength, but larger than the coronavirus particle, or the coronavirion, as I should say, be optically correct. So we have the active domains. Let's say there is a 4th one, but I'm not considering for the study. And we have done basically the matching of the sensor for each of the proteins that we have in our range. For the first part of the study, basically, what we did was we functionalized a Ball resonator. For only this type of sensor, we have also here a Bragg grating, because a tilted Bragg grating, because we are still not being able to extract the spectrum completely. So we had help from another sensor. But as you will see at the end, we will not need this part. And we performed the same functionalization. We did several replicas of the sensor, one for each protein under detection. That's the spectrum of the sensor as it look like. It's different from the previous one because we have these additional cladding modes. But for a moment, forget about the cladding modes, and as you can see, the spectrum has the typically irregular shape of Ball resonator. So we have again a random spectrum. This is the characterization for refractive index, and this is how the sensors were functionalized. So we have specifically selected the L1, A27, A33 proteins. We have selected antibodies binding to each of them. We have done the ELISA tests and particularly the Western blotting, so we could really be quite specific about quite sure about the specificity of each of the antibodies in this study. And then we perform the detection. So this is the spectrum, for example, that we observe for the L1 protein, starting from a very low concentration and moving up. And we have again the same pattern that we had for the cancer biomarker. So that's of course confirmation. We were quite sure about that because the protein is small compared to the wavelength. So as a matter of fact, the effect of the protein on our sensor is very similar. So we were quite sure about that. This is when we actually perform a detection. So we did that in PBS and we did that in a serum. In an undiluted serum, which of course is a quite large, quite hard type of detection scenario, we still have a quite good performance. For one of the proteins, we could really mark down the limit of detection to very low one. For the other ones, we don't really have such a clean limit of detection, but still a very good one because we're talking about few picomolars in considering that this is an undiluted serum, so that's quite tough work environments. Actually, this is 827 and 833, we just didn't render very well. And this is a kinetic of our measurement. And as you can see, with the 3 minutes step between measurements, the data are very stable. So that means that every concentration change, we went in constant way here, but in reality is actually, choose a different one, is nearly instantaneously rendered on the surface of the fiber sensor. So the detection time, we can say that is quite instantaneous. And here it comes with the analytical limit of detection that were performed. So in PBS, of course, they're very low. PBS is quite ideal. When you actually change the medium, they're still quite good one. So that's a very attractive type of sensor. So the proteins are recognized very well. And we could test the specificity, both internal, where basically we compare the antibodies bind to each of the protein to the antibodies bind to the other one, and with other external and non-binding controls. The specificity actually is quite good. But that's the detection of the protein. Can we detect the virus itself? So the idea is, if we can see the protein, can we see the whole viral particle? So we actually tested the same concept, except this time we expose the fiber to the virus. What we did actually was, as I said, now the critic is gone, so we have done a few years into development of this, so now it's more reliable in detection. The functionalization that we have made is exactly the same, and we have tested 2 antibodies. One that is the L1 protein, the best performing one, and one that we synthesized from rabbits. So rabbits were inoculated the virus, and we got the antibodies, synthesized and purified those antibodies, and we could compare the 2 results. That's our setup. So again, the hardware, that's not our lab. That's because we made these measurements in one of our partners in Milano, in Italy, where we actually have a lab with the appropriate biosafety level, and where basically we could place right here our ball resonator. And we have investigated several of them. So that's how they work. First, this is the one for the L1 protein, the best performing protein that is found on the surface of the vaccinia virus. And yes, we have again the same pattern. So when you actually start from an enriched PBS right here, back into all the virus concentration, you see that there is a quite visible sensitivity. And yes, this is again a random spectrum. So each time the spectrum will look different. So there is a sensitivity that is appreciable quite here, really quite large. Notice that the change that we have going from 1000 to 10 to the power of 8 plague forming units is quite large. We're talking about 6 dB. That's quite large change of signal. And that's actually how the dynamics of detection works. The data are sampled every 3 minutes. So notice that the response is quite staircase. So that means that after reaching a certain concentration, the system remains stable. And when detecting the next one, the system changes quite rapidly. And that's how it works with the other type of antibody. So again, the spectra are quite random. We select one part to analyze the data. And when we point it out here, we have again a decrease in the spectrum. This time is even larger because the sensor has a larger sensitivity. And again, we have a detection here. So yes, the sensor sees the virus. That's a quite reliable result. And as you can see again here, we have a response of the sensor that is again quite as a staircase. So we compare different case scenarios. The sensor that has the L1 protein as a target. That is on this curve right here, the response for each of the concentration. And it's almost linear with some saturation. That's quite good because the sensor is detecting an enriched PBS. So it follows the same trend of the protein. That's actually quite reassuring for us. We have the detection with the rabbit antibody right here. It's not that dissimilar. If you compare them one-to-one and normalize by the sensitivity, they're actually quite similar. But also, we have a similar response when we introduce a control as well. So that's another particle, the herpes virus, that we use as a control. Notice that the herpes is an even larger particle than the vaccinia virus. So as a control in general, if the system is not specific, you will start to see some problems there. But the detection actually has a quite good level of matching. So the data are pretty good match. And this is the specificity of the chart. What we actually can see, these are multiple sensors, right on the left. The first three are fabricated with the anti-L1 functionalization. And on the right, they actually are fabricated with the rabbit antibodies. The main problem that our protein-based sensor actually solve is that they are quite specific, because they do have the classic pattern, not with the same sensitivity, but in terms of control, the green curve actually is quite low. What we don't really like is that the ones synthesized from the rabbit and therefore targeting the whole virus, they seem to perform worse in the terms of specificity, probably because the actual method, antibody extraction and purification, might actually point you to select an antibody that is broadly binding rather than one that is specifically binding. So that's actually the result we have so far. Of course, it's still undergoing, but it's quite reassuring. So there is a possibility of detecting viruses using these fibers. And based on what I said before, we can make that parallel, we can make that urban, we can make that into wastewater, we can make that into gray waters, and we can make them multiple environments. So let's draw some consideration and conclusions. First of all, the detection of pandemic viruses in liquids, wastewater, gray waters, but also sputum and saliva and air, such as breathing, is a key asset. And I think it's a biosafety problem that will come up in the future. Moving from the detection of the protein to detection of the virus is working. So we now have the data to back at least this part up quite confidently. The question is, the protein available for binding is much smaller than the total protein. How does this affect the virus detection? And how is that depending on the virus that we have in target? That's a question that we have to address still. Multiplexing is the key. You have a virus with 4 proteins, you want to have 4 sensors that detect it. And then you would like to have 4 sensors in another detection to detect it. And then you would like to have 4 sensors in another location to detect it. And then you would like to detect it. So you see that the number of sensors that you would like to ideally have start to scale up. And in a real environment, how the duration of antibody, the temperatures, how do they affect? We don't really have a clear answer. We don't know exactly how long antibodies will survive at cold temperature, how long they will survive at room temperatures, how often we would have to replace the sensors if we were to make a real-time detection. But we know that the sensors are low cost. At least this we know, because the functionalization is a very standard protocol that we can repeat it over time. And we usually fabricate many sensors at the same time. And the fiber optic sensing unit that we are using, they are very cheap. They're based on either telecom fibers or a custom fiber that has the same properties of a telecom fiber. To give an idea, the cost of a telecom fiber is about $8 to $10 cents per meter. So we're talking about a quite cheap type of platform. So in conclusion, optical fiber sensors can detect a plurality of analytes at extremely low concentration. They have a minimum Z4 factor, high performance, and a lot of other nice perks that makes our job actually quite a little bit easier. Minimalistic approaches improve the affordability and scalability while allowing multiplex detection. But we have to work with random spectra. So if there is somebody who wants to tackle the approach of how to work, not with the common spectra, but with random spectra, of course, that's an open issue. And we will welcome, of course, some suggestion and collaboration in this regard. So the virus detection in wastewater, in gray water, can it become a watchdog for deadly viruses or pandemic viruses? The idea is that we have to now the technology to make the effort. So that's actually my hope. I just wanted to thank some of our collaborators that actually are all around the world. Several of them are based in Italy, some of them are based in Belgium, in Spain, in Portugal, in France, in the United States, and in China, that actually have really supported us to our work. That has been a quite collective effort rather than first. So that's the end of my talk, but of course, if you have questions, I will be happy to address them. Thank you.